Cytokines are the signaling molecules in the immune system. They activate it, they turn it on, and we have made drugs that can actually use the natural signal that's already there of a cytokine called interleukin 15 and bring it in and add it to the ability of a drug like Keytruda to start to activate the immune system. Cytokines are nature's own way of regulating the immune system. So very often cytokines are able to activate a particular group of immune cells. If you have a viral infection, for example, the cells that are infected by a virus will start making interleukin 15. They actually display it on their cell surface. In that case, the cytokine functions as what's called an alarmin. In other words, it's the alarm system of the cell.
Hello, David. Thanks for joining us today in Scouted by GreyB. I'm very happy to meet you. Really excited to have you here. So how's it going? How's everything?
Everything's good, Shabazz. It's a beautiful day and ready to get going. It's early in the morning here. So you're my first meeting.
That's great. I guess then I've caught you into a very good mood. So let's go ahead. So David, before I jump right into it, can you share any recent news, any exciting things happening with Reverb Therapeutics these days? Anything that you haven't made public yet, or maybe you'd like to share with me?
Yeah, so Reverb Therapeutics is in Vancouver, Canada. We were just at Web Summit, which is one of the biggest meetings, the biggest tech meetings, where for the first time there was a session specifically on biotech and more specifically biotech in British Columbia and Canada. We're able to let people know a little bit about our ongoing research and about our first drug candidate, which we'll be nominating at the end of this summer. So we're able to now just let people peek a little bit behind the scenes in the progress that we've made.
That sounds very exciting. So, David, just for our audience, can you, in something that a normal person like with me will also understand? So, what is Reverb Therapeutics doing in oncology?
Shabazz, first, I found out you are wicked smart. So let me just make sure that other people understand it as well. What we're doing at Reverb in oncology is we're making drugs that amplify the effects of the immune system and allow the immune system to better both detect and attack cancers. As companies often do, we're standing on the shoulders of giants. 10 years, maybe 15 years, there's been a huge revolution in cancer treatment with a set of drugs called ICIs, immune checkpoint inhibitors. And what they're doing is they're allowing the immune system to detect the cancer again when the cancer tries to hide itself. And the best known drug in that class is a drug called Keytruda, which is, I think, also the biggest earning drug in the world. And that drug has made a huge difference in lives. So the estimate is that about 50% of patients who have cancer in the United States are eligible for treatment with Keytruda or a drug like it, which is amazing. And it's revolutionized treatment, for example, of non-small cell lung cancer. But regardless of those improvements, what we started to focus on at Reverb is that for many of these patients, it's a temporary respite. This is not something that will give them the life that we'd love for people to have if they have cancer. And what we do know is that there are other parts of the immune system that are very important as well. So we are bringing in another part of the immune system and specifically molecules called cytokines. Cytokines are the signaling molecules in the immune system. They activate it, they turn it on. And we have made drugs that can actually use the natural signal that's already there of a cytokine called interleukin 15 and bring it in and add it to the ability of a drug like Keytruda to start to activate the immune system. So what that does is it makes the immune cells even better able to not only detect the cancer, but also to multiply and then attack it more proficiently. So our lead program, which we call AMP-01, amplifies specifically the effect of PD-1 inhibition, which is the effect of Keytruda, by bringing in interleukin 15. So that's what we're working on.
Okay, so I think what I understood from it is that unlike many other cancer therapies where a new foreign drug is introduced into the body, you're leveraging body's own ability to fight cancer and immune system, and you're basically modulating that and basically helping it detect cancer and produce more cancer fighting cells. So have I got it right?
Yeah, exactly. So the way we talk about it is it's redirecting the efforts of the immune system to really make it concentrate on this particular bit. Your immune system is amazing. For most people, somewhere in your body, there's a cell that's doing something wrong, that's precancerous or even cancerous. Your immune system detects it and removes it. So very often, for most of us, our immune system is able to do this. In patients with cancer, very often the immune system is just under that ability to go and control tumor and tumor growth. And we're hopefully helping it to now amplify that effect and redirect it so that it's more specific to the tumor.
Okay. So you mentioned cytokines, and I think when I was doing some research, I found there are like multiple types of cytokines, and I think Reverb is using interleukin 15, which is like very specific type of cytokine, right? So I was curious about it because I couldn't, no matter how much research I do, I can't reach that level of knowledge that you might have in this space. So just was curious about like what makes cytokines so appealing for cancer therapies compared to other immune modulating therapies out there.
Yeah. Well, cytokines are nature's own way of regulating the immune system. And there are hundreds of different ones. And we give them numbers and different names. And in this case, cytokine comes from the Greek verbs to move cells. Cyto is cell, and kine is movement. It's really to be able to move cells. So very often cytokines are able to activate a particular group of immune cells. And interleukin 15 is special because it's nature's way of calling in two cells that clean things up. So if you have a viral infection, for example, the cells that are infected by a virus will start making interleukin 15. They actually display it on their cell surface. Now, in that case, the cytokine functions as what's called an alarmin. In other words, it's the alarm system of the cell. And interleukin 15 then activates a set of cells called natural killer cells, which are innate. In other words, they'll attack anything they see. And it activates a group of cells called cytotoxic T cells. And cytotoxic T cells react to a particular antigen, to something that's sticking out, maybe a piece of a virus, to go and attack that particular cell. And NK cells just attack everything that's there. And if you have NK activation or natural killer activation, as well as cytotoxic T cell activation, you can very efficiently remove cells. So what we've done is we've really hijacked that natural system of cleaning out a tumor, an infected cell, by putting it on the tumor. So we're in a similar way, essentially displaying interleukin 15 in the context of the tumor, trying to get these NK cells and these cytotoxic T cells to come in and to remove the tumor. Other cytokines have other functions. They may activate B cells that make antibodies, or they may actually calm down inflammation in a particular case. So cytokines can have many different tasks. So all of these molecules are very specific, and interleukin 15 has this amazing ability to bring in these two groups of cells that can clear out the tumor.
Okay, I think it makes perfect sense that use your body's own strength to fight a cancer, and instead of introducing a foreign. But now this sounds like a perfect scenario. So my next question is around like, is there a, I would say, drawback to this, or basically why has no one else figured this out?
So first of all, we are not the first ones to figure this out at all. So I got together with my co-founder, with Surjit Dixit. Surjit and I met about 3.5 years ago. And actually, this idea of bringing cytokines in together with inhibiting PD-1, which is the signal that Keytruda inhibits, is not new. We are not the first ones. That biology is out there. But usually the way people do this is they take a cytokine like interleukin 15, and you'll hear me call it IL-15 for short. And there's another one called interleukin 2, which is very similar. And they will sort of attach it to the drug, to the antibody. So what they're doing is they're taking one of these cytokines and turning them into drug. And these are called cytokine fusions. The cytokine is fused to, in this case, the antibody that can inhibit PD-1. The problem with that is that cytokines are really meant to act locally and briefly. Like if you turn on the alarm, you don't want the alarm to be turned on everywhere in the building. You only want it to be turned on where the issue is. And you also don't want it to be on all the time. Your ears go deaf, you stop reacting to it. And similarly, cytokines are meant to act locally and briefly. The moment we turn them into a drug, we need to administer them to the whole body systemically, and drugs work over a prolonged period of time. So it's a little bit like just turning on the alarm everywhere. And what that does is it causes very often what's called systemic toxicity. So the whole system everywhere now reacts to that cytokine. And if this cytokine is very pro-inflammatory, like interleukin 15, you could see that that's bad. You don't want inflammation everywhere in the body. You really want to just turn it on. So the first issue with these cytokine fusions is that one way or another, people have to smuggle them by so that they're only active in the tumor. It's very hard to do this. The second issue that you have with these cytokine fusions is that they're very often just close enough to the real thing, but a little bit different. You have to fuse them. Very often people have to stabilize the protein, so they have to change a couple of things. The immune system says, hey, that's not the real one. And what that does is it gives an immunogenic response. In other words, your immune system starts to react to the drug and actually inactivate the drug. And that's very common as well. The third one is, and you can sort of hear me talk about this, is these are very complex molecules, very, very difficult to make. And Sergeant and I had this idea to not make a cytokine fusion. So what we're actually doing is we're saying your body constantly makes cytokines at low levels. It's always there, and it just keeps on making them. They generally have a very short half-life. They're very short-lived, and then they're removed. Why don't we capture that with our drug? Our drug is called something called a bispecific antibody, rather than trying to make it. And what we do by having one of these drugs that just captures the cytokine that's already there, is that first of all, we don't dose an active cytokine into the body. So we don't have this issue of systemic toxicity. We capture the natural cytokine that's already there, which means that your immune system already knows it, doesn't react to it. And the molecules are much simpler because they look like bispecific antibodies, which are very well-known drugs. So what we bring, our innovation, is really the way we're tackling the problem rather than the biology of the problem, which is what we've just discussed. So the technology behind it is different. And the technology we've called amplifier. You can use this in a variety of ways. You don't have to only use it for interleukin 15. You can use it for almost any signal in the body that you want to concentrate in a particular place. So we have versions of molecules that actually help the immune system to calm down in particular tissues. For example, when people have colitis or when they have psoriasis, or when they have rheumatoid arthritis. So we can play with all of these signals and sort of just give them a boost in a local setting to overcome the issues that it's just not there enough to control that particular disease process.
Okay. I think it makes perfect sense. And I think the differentiating factor you have shared, so it's very strong. It makes sense that, and that instead of, administrating a drug externally, which might cause toxicity across the body, across all the organs, you have to keep it localized, which makes perfect sense. So I think what I also wanted to know is about I guess with this, after hearing you, what I'm able to figure out is that any drug that is using external cytokines, introducing them in body, they also run into this issue of manufacturing them and making it very specific for the patient's body, right? So am I right to assume that problem is also solved since we are not manufacturing them outside of the body and we're just helping the body produce them?
Right. No, absolutely. So we do solve that issue. And sometimes there are drugs that people are developing that have used this fused cytokine where they've made a drug the way I described, and those are moving into the clinic. And in cancer, very often our drugs cause some problems, but the benefit outweighs the risk. And so I'm very excited by the prospects for patients with cancer to get one of these drugs. And there actually right now are a number of them that combine also the inhibition of PD-1, which is this Keytruda signal, with blocking another cytokine that's called VEGF. VEGF is something that makes blood vessels grow. There's a drug from Summit Therapeutics that is in phase 3 trials that just had a little setback, but looks to be a really great way to block again both this inhibitory signal for the immune systems to allow it to start to recognize tumors, as well as to stop angiogenesis, to stop the forming of blood vessels. And yesterday, BMS acquired one of these drugs. So these are called PD-1 VEGF drugs from BioNTech, the German company that also developed the vaccine with Pfizer. So it's a very interesting space, and we see a lot happening here. And we wish everybody a lot of success. And I hope they overcome these issues. But we feel we have a better way to address this in a way that will be safer and more tolerable for patients.
So far, have you faced any challenge in proving efficacy, maybe, or how well this has been working so far?
So here's the problem. Until you are treating patients, you're treating mice, and mice are furry test tubes. We have, and I have, cured so many mice of cancer, and it's meaningless when it comes to people. But we are not furry test tubes. We also are very, very different from each other. And these mice, they're inbred, they're very consistent. And generally, what we call a tumor in a mouse is actually a cell line that grows under the skin and very rapidly grows. It's, I guess, in some ways you can call it a tumor, but in other ways it's very, very different from the natural disease in humans. In those settings, we do better than drugs like Keytruda. We're able to control tumor growth better. Even when Keytruda fails, our drug actually works very well. And we think this is meaningful just from the perspective of we can show that this is because we've added IL-15. We've added this signal of interleukin-15. And we have a strong set of pieces of clinical evidence that in humans, that combination should work very well too. But until we've run our phase 1B, we're just starting to raise an A round. So we should be there in about 3 years or so. Until 3 years from now, whatever I tell you is hopeful speculation.
I think being hopeful is the key to reaching that goal. And I guess if things are working on trials, I wish you good luck. Everything will work out. Not that, it means much coming from me.
No, it does. So one of the things you learn in biotech is nobody should be joyful about drugs failing. This is such a hard business, and it's so difficult to help patients that every piece out there is a piece of success, and we need everybody's shoulders under this. I would say the bravest people in drug discovery and development are the patients who take that drug very early on in clinical trial. They're amazing, and they should get credit for the work that they're doing, which is use their bodies to help other people. We're using our minds and our hands to do the same thing, and your good wishes are incredibly helpful in helping us move forward.
Great, I'm happy to hear that. So, David, I think I understand the science behind it a little, and now I'm also curious about, now that I also understand where we are in terms of clinicals and basically testing, so just want to understand. Now let's move towards a little bit of marketing or market side and scaling side of things. So how has been things working in case of moving towards a next round of funding, or I would say showing more market viability of AMPO1? So can you share a little bit more about what kind of opportunities you are looking for and how things are going?
So we're planning to raise a $55 million, what's called an A round. The next round, which should get us to a very meaningful inflection point for the company, which would be the phase 1B, where we actually show that the drug works in a variety of tumor settings. Raising money right now is incredibly difficult. If you look at biotech over the last 40 months, public biotech companies have lost an aggregate 90% of their value on the stock market. And that means that if somebody has money to invest, they can either invest in me, we are privately held, and it takes at least 3 years until we get to a meaningful inflection point where there would be another round of investment, or somebody might be interested in acquiring the company, or they can invest in public companies that are very, very undervalued right now. And actually, for a quarter of them, they have more cash in the bank than their market cap. So they have negative enterprise value. So let's imagine that there's a company out there there are companies like this that has 500 million in the bank, but the market's valued them at 50 million. And somebody has a $5 million check, they can buy 10% of the company and functionally 50 million of working capital for 5 million. Or they can invest in me, and that 5 million will just be 5 million. I'm going to need to run my next trial. And by the way, you're locked up for 3 years because I'm privately held. You can't trade stock in a company like mine. Whereas in these public markets, obviously you invest, and if things don't go the way you want to, you can sell again. So what that means is right now, privately held early stage companies are having a very rough time. However, I very strongly believe that good ideas and good science will find ways to get funded. And we definitely are seeing interest from large pharmas and from venture firms in this particular idea because it could be transformational. And I think it's the only thing that we can do right now is come with transformational ideas in early stage venture and see if we can push them through.
Makes sense. If I had $5 million, I would definitely invest.
Oh, yeah. And you're more than welcome. Even $5, we'll take it.
Yeah. Again, thanks for sharing all this. I think apart from market and of course, the challenges that biotech startups are facing about funding, how do you see any regulatory challenge also coming up in future, like for Amplify our platform?
And not necessarily for the platform per se, but I think we're going into a lot of geopolitical uncertainty, and in terms of the role of American regulatory authorities. Until very recently, the FDA was the gold standard for the world. And we think that will continue, but we don't know for sure. The other thing that's changing is the attitude of the FDA towards safety studies that are done in animals. So before you enter into humans, you want to establish that your drug is safe. And we want to definitely make sure that before we dose the patients, we are personally convinced that this is a safe drug. Otherwise, we wouldn't administer it. And although the FDA has said we want something else, we want people to think about other ways to establish that safety, that pathway isn't completely clear yet. It's not clear what would be acceptable. There's a lot to figure out in the short run, and regulatory uncertainty may lead to people looking at other places to go and get their drugs registered. So Australia is a common place. You can go to the EMA in Europe, and you can go to England, and you can go to Canada. And first get your drug registered, your clinical trial registered there. And then maybe look for what's called an IND, an investigational new drug application with the FDA a little bit later on when you already have human data, which should establish safety a little bit better. So that switch we're in the middle of, and it will be a very interesting time. It will be very interesting to see who else in the world will take the lead on the regulatory innovation.
Okay, makes sense. I guess then what I also want to understand is that since this is a very new area and you're essentially coming up with like entirely new science to solve this problem. And of course, I'm not saying that you're the only one, as you have already shared, but I think this space requires as much participation as it can get. So you're already on that track. So David, from intellectual property perspective, just wanted to understand basically what's your strategy of protecting all the new things you're making? Are you going with like, of course, pharma research is super difficult. It takes a lot of time. It takes a lot of money. So basically, what's your strategy behind that?
Yeah, no, it's a great question. Intellectual property is a very interesting. It's sort of always implied and never discussed. So it's a great question to ask. But for me, intellectual property comes in three layers. The first one is the big idea. And for us, the big idea was this redirecting endogenous cytokines. And that has two parts. The first one is making antibodies that can engage cytokines or drugs that can engage cytokines in such a way that you stabilize them, that they remain active and able to engage their receptors. That turned out when we looked at the literature that that's not a novel idea. People have tried to do things like that for almost 40 years, or at least made observations like that. The second part was take a cytokine and then redirect it and attach it to a cell. The fused cytokine molecules I described earlier do exactly that. So that part isn't new. But this idea of redirecting what's called an endogenous cytokine, a cytokine that was made by the body, was completely new. So nobody had really put these two ideas together. So the first thing we did is we established foundational IP on this particular concept. And we filed that now, and you can go and look it up if you want to. We're actually broadening that set of patents as we get more specific insights on how we can go and protect this idea in general. The second level may be around things that are specific to a particular cytokine or a particular target. So for example, we now understand very well not only where to engage interleukin 15, because on the molecule, it will depend where you bind it, but also how to bind it so that it actually has the effect you want to. We actually want to modulate its activity. And I think you used that word before. That's a second layer of IP that's around targets specifically. Now, the third layer of IP is always around the composition of matter. It's actually the order of the amino acids that go into these particular molecules. And in general, you want to apply for that as late as possible because you want that period of exclusivity to run as long as possible when you're there. So if you have a really good first layer and second layer, you can wait filing on the specifics of your molecule until you have to disclose it, which is generally early on in clinical trials. So that's the strategy in order to go and make this work. And you can now follow this around every single drug. The reality is that small companies like Reverb cannot really exclude others because we can't really have patent fights. We need to be very, very well funded to send lawyers out to go and discuss with other people. However, people who acquire us may be ready to have those patent fights. So at a certain point, the natural history of companies like Reverb is that when we're successful, 99% of cases will be acquired. And the acquirer, if you've done a good job establishing a patent base, then will value that because that allows them to now exclude others from this particular space.
I think, yeah, this is one area where I have some knowledge since because of GreyB's work. I totally agree with you. IP And having patents on your innovation, it's like, I would say, one of the selling point for a lot of companies. And definitely, and I think it's also not about preventing others from doing the same. I think IP, that has like this image has been built over time, but I believe IP is more of incentivizing company like to innovate and file more patents. So of course, I think somebody else can also work on the same technology, but at least you will get credit for it, and you're not like really gating this technology simply by having a
Right.
patent.
And in many ways, patents are a tacit way of collaborating because we're teaching each other in patents. And I think and I hope that, like I stood on the shoulders of people who did fantastic research before me, people will use my ideas or our ideas and stand on those and be able to go to the next step of ideas. And IP is a wonderful way of making sure that you're very clear about what your idea is and what you thought of and what's so unique about it.
Definitely. And I think you're already on the right track. You have very good strategy in place. So again, that's great. So David, with that, that was all the questions I had, and what I should say, I learned so much from you over this call. And I guess just one last thing I'd like to know is that you have like amazing experience in this space. You have been like very higher position in your previous companies as well. And now that you're like working in entirely new space, working on new solution, and this space is highly competitive, even though your solution is unique, right? So, but oncology is like, it's a crowded space. So, what will be, I would say, and of course, this is a feeling every founder might get, that maybe this is not going to work. What if we fail, or what if we don't reach where we want to reach? I'm safe to. I've talked to so many founders. Everybody faces this question, and this is something that keeps them awake at night. So, how do you deal with that? If you also go through the same emotions?
So this is the fifth time I'm a CEO. I've added ideas to companies. This is the first time that it's really something that came out of a deep discussion that I had with my co-founder early on, from my brain and his brain together. And you're right. It's very hard to distinguish who you are from your idea. And so I think what helped me is actually meditation. And one of the things I do there is I let these worries come in. I realize they're there. And I give them their own place, but I separate my worries from me. Otherwise, I can't live, because there's so much uncertainty in what we're doing. The chances of failure are so high that I wouldn't be able to get out of bed. Meditation is a big part of my personal strategy to make sure that I can function day to day. The other thing is having other things to focus on in life. I like to work, and I work a lot. I also like to cook. I spend a lot of time with my wonderful daughters and my wife. And that also helps me, because when I'm there, very consciously say these worries can be put aside because they're not for this particular moment or this particular interaction. So I think it's meditation and having other things to do in life that got me here.
I guess
Can I
before
say
that,
one more thing, Shabazz? I think every good CEO or COO or CSO I've met in biotech has been in therapy at some point. These are very stressful jobs, and there's no shame in saying you should take care of your mental health in all of this.
yeah, I agree. And I think I have talked to a couple of biotech founders as well in the past, not necessarily in this space, but everybody is working on, sometimes in gene therapy, sometimes in cancer research, but on testing side. So I see them share the same stress of working in biotech. Biotech, I think by nature is a very stressful. You're working on something so tiny but so impactful, and there's always pressure of moving to the next step and finding your way around it. So definitely makes sense. And I really like your approach of looking at other things. It's like a Buddha approach, right? To life. So it makes perfect sense. So with that, I think, David, that was like all I wanted to know today. And thank you for
Thank you,
sharing
Shabazz. It was a
details.
pleasure talking.
Yeah, same.